So good morning, everyone.
Good morning, sir.
Good morning. Yeah. So respected all the participants, I am Dr. Kiran Sree, the coordinator of this faculty development program, and hope we are enjoying this all the 3 days of faculty development program. And this is our fourth day, where today I thought of having a session on deep learning, but I changed the topic like. How can we design an expert system by integrating deep learning with cell automata? If you see the title of the workshop that we have chosen, you can understand that the title uh, is biased towards creating some research ambience in interdisciplinary research pertaining to artificial intelligence and robotics. That is the reason for the last 2 or 3 years, I am working on applying these deep learning models to various parts of, to some problems in bioinformatics. We have collaborated with EEE department of IIT Madras so that we are applying some deep learning models for power systems and control. And we are integrated with some civil department in our institute where we are applying our deep learning models for civil department. So I understood for the last 20 years when in the academic arena, I understood interdisciplinary research is of highest significance. That is the reason the title and whatever discussion that we are doing for the last 3 days and the fourth day also, uh, all this will create some ambience towards interdisciplinary research. So the title of the talk is very simple. Before getting into the title and discussion, I wish to just introduce myself because I'm the coordinator of this event. I am Dr. Kiran Sree, and I have my BTech degree in Computer Science and Engineering and ME from Anna University in Computer Science and Engineering. And my PhD was in the area of artificial intelligence and deep learning. And I have published almost 96 papers in various international conferences and journals. And in these 96, two are Sciendex journals and 20 are Scopus Index publications. And I have filed 4 patents on various aspects of integrating deep learning with IoT. And one of the patent, most probably, I may be awarded by the end of this month, or the most probably starting of the next month. And I have authored 6 textbooks on various aspects of deep learning and IoT integrated with artificial immune system and so on and so forth. And I was a BOS member of Vikram Singhpur University for almost 2 years and SV University for 1 year. And at present, I'm heading the Department of Computer Science and Engineering at Sree Vishnu Engineering College for Women, Bhimbaram. So the title that I have taken for this talk is how can we design an expert system integrating two techniques of artificial intelligence. Cellular automata is a technique in AI, and deep learning models are the upcoming models that most of the people know, like whatever real-time problems now are existing. We are trying to propose deep learning models for all these problems so that we can get more accuracy and we'll get the output faster. So the same nomenclature that we have done very recently, I have published in Sciendex paper. The discussion that we are going to have today will be to some extent will be relating to that paper and towards developing an expert system. So the motivation, I have organized the slides like this. I start with what exactly is the motivation that I have in choosing this topic. And then my interest is to create some interdisciplinary research in this aspect. So that is the reason the more parts like whenever I wish to do some research in power systems and control, what type of domain is different and what is that we need to do. So today we are going to discuss something like we have taken some 2 potential problems in bioinformatics, interdisciplinary research. Whenever you are proposing interdisciplinary research, what things we have to do. This type of discussion we'll do, and at the end of the day, if there is a chance, we'll also discuss what type of deep learning models will help us and what type of deep learning models will not, like what deep learning models can be integrated. And now, recently, if you see the recent advancements in deep learning, we have a model called as ternary neural network, which is an advancements of deep learning, where most of the people are using this ternary neural network in combination with CNN and RNN to have a potential and robust classifier for more accuracy and all the other things. So the motivation behind the particular talk is something like we have an expert system called as mycin, where the mycin has almost 56 different type of versions. Initially, a mycin is an expert system. What it will do is it will take the input in the terms of some symptoms, and it will give you the disease associated with that particular symptoms. But slowly, what does the mycin is doing is mycin is taking the genetic information of the patients through the blood samples and the analysis that were done, and it is going to predict something. Is this person, if the data is that if this person is prone to some diseases or not, and if he's having some six fingers in the hand, what is the reason for that? That type of biological analysis now the mycin is trying to do, and it is doing. If this mycin link was already there in WHO. So the basic motivation that I found for this particular interdisciplinary research is like mycin. I have studied various versions of functionalities of mycin. And what is that I have done at the end of the day is I have taken two or three major objectives of that mycin, and what is I thought of applying my own technique in solving the problems that mycin has already done, and what type of data sets that mycin has taken, and we have also taken the same data sets. And the reason that the challenges we have, the objectives I will discuss, but this is what the motivation that I have for mycin. But the interesting fact that we understood from mycin is initially mycin used to interact with people in the terms of text. Now mycin is interacting with people in the terms of voice, and so on and so forth. Now, if you develop some AI system, and the minimum necessity that AI system requirement is to interact with the people in the terms of text. The second major requirement that AI systems or AI apps must have is they need to interact with systems with respect to voice. And now you have many expert systems like Watson and other expert systems. They are also processing videos for addressing various issues. Like if you see the last month paper on Hindu, you have an article on crowd counting systems, where this crowd counting systems will analyze the videos and will tell you how many people are there. Apart from that, they will also tell you how many people are having abnormal actions based on the training that was given, who can damage the infrastructure and so on and so forth. So likewise, the intention that I have for this particular presentation is I have gone through many expert systems, and I have observed the behavior in which the expert systems are working, and I have seen the various objectives of the expert systems. And what is the thing I have done is can we develop an expert system by using deep learning augmented with cellular automata, because both are artificial intelligence techniques, and most of the people from, if you have an IISc Calcutta where cell automata lab was there. So most of the researchers are working towards cell automata, and many of the people, like almost 40 to 50% of majority of the research is going on artificial intelligence in the terms of how do you use these deep learning models for addressing real-time problems and societal issues. So the same mindset that I have, and I have started taking this two potential problems in bioinformatics, where I understood like mycin has also already also in background it is giving the outcome in the terms of processing these two regions. I'm repeating this again, and mycin is an expert system which is going to give some outcome in the terms of whether this person is prone towards cancer or not. Whether if he's having six fingers in the hand, what is the reason for the six finger? Whether see when he is suffering from chronic disease, what is the reason for having the disease? This type of outputs your mycin can give. And if this mycin has to give this type of outcomes, what is the internal structure that it has so that it is giving this type of inferences and predictions in future. So we understood after that, like these two potential problems that were there in bioinformatics were behind this mycin. So we thought of having our own solution for addressing this protein coding region and promoter region. Then we started to integrate we started to find algorithm to predict these two by using integrated algorithms. This is what the entire agenda that I have for today. So I said the motivation that we have for this lecture is very simple. Like we thought of applying this when we thought of applying or pursuing our interdisciplinary research, and we thought of integrating artificial intelligence and some other department. I thought of having an artificial intelligence technique that can map or that can have the same framework that we have. And I understood, like after reading this bioinformatics various papers and implementing some of the real-time projects, I understood that bioinformatics input is different, and the particular domain of operation is very difficult because whenever we wish to process this DNA sequences, and all these genes will be there in the part of DNA, and DNA is a minute part of the body, and you need to identify the locations very difficult because you need to identify a minute part of your cells. So all these challenges are there. Then what is that we done is whether we had done a feasibility study, like whether the cell automata, whatever we have, is applicable, or whether the framework that we have for this bioinformatics problems can be applied on the cell automata or not, or is cell automata is suitable for having this on bioinformatics or not. This is what we have done. So for this, we have approached a Coral lab from IISc Calcutta, where Dr. Pradipta Maji was the in charge of that lab. He is a prominent person in the area of artificial. Pradipta Maji is what he is the in charge at that place, and we have approached him. I personally worked in that lab for almost 6 to 7 months in 2011 and 12. So we approached him whether this technique can be applied, and after that, after a few years again, we have approached him whether cell automata integrated with deep learning will work or not. And then, because he's already done some research on interdisciplinary areas of applying the cell automata and deep learning on various aspects of power systems, bioinformatics, and particular problems related to ECE, then he said the framework suits, but we need to choose a appropriate a kind of cell automata which can help you to find the potential classifier. So we felt we felt that we have done the literature survey and we have done the feasibility study, and we felt that appropriate cell automata model, whether it is a nonlinear cell automata or a hybrid cell automata or integrated cell automata, that has to be selected, which can map with this. But if you see the agenda that I have kept here, the agenda looks it's very simple. But what is that we have done actually in this research is we started with two problems, but the intention that we have the behind developing this particular work is like, can we develop an environment, or can we develop a classifier which can solve majority problems in bioinformatics? The same thing we are doing in power electronics also. When we are getting some various problems in power electronics from EEE department, we started with a single problem. Then we again understood that this problem is related to some other problem. We have taken two problems, and slowly we have designed a classifier which was in using CNN in deep learning, convolutional neural network with Terra neural network, and this combination and the framework that we have developed is suited for at least 10 problems in this power electronics and pumps. So the same intention that we have here, even though we have taken two problems in bioinformatics or the problem that mycin has solved. But the intention of developing this classifier is this classifier framework should be fitted in such a way that at least at 5 to 6 open problems in bioinformatics can be addressed by this tool or classifier. So slowly what will happen is when many of the issues in some domain are done, so we can have a tool like this. So that is the reason we have a dedicated web server which was maintained by the Department of Computer Science and Engineering at FCCW for finding whether some DNA sequence is having protein coding region or not, and having promoter region or not, and what is the time that it has taken. So the time taken that to predict this protein coding region, what was that we understood is 0.7 nanoseconds. And what is the accuracy that we are claiming, and what type of data sets we have taken, and from where it has taken, and every day, every almost 7 days, we are updating that web server so that this can be compared with the existing ones. So if you just go to the next slide, like what will be the objectives of research? Many objectives are there. I said if people are suffering with chronic diseases, what can be the reason for having that? Whether the disease is inherited from father or mother? Just a minute, we will just continue. Okay. So in between, we'll just continue. But see, for the last 3 days, we are maintaining the attendance in the morning and evening sessions. And there is a mail which was received in the morning from ATAL that attendance has to be taken strictly. That I can just forward it to your group also. Like every participant that was participating in this any FDP of ATAL has to at least he or she has to maintain 80% attendance, and attendance requirement is mandatory. And you know that tomorrow you have a multiple choice exam. Uh, uh, for almost half an hour, and you need to get at least 60% marks. I personally participated in 5 or 4 at ATAL FDPs, and we have done I have done that, and the attendance to some extent is not promising at this moment. And at least 80% of the attendance has to be there. If you have any problem in attending these FTPs, please inform us, because every day we are updating this portal. So at the end of the day, if you just represent that I was there in the second day and I was there in the first day, we cannot do anything, because every day I am updating the portal with the attendance that I have for every for that day. So just will continue, like objectives of the research that we have here is like if some person has given I have taken a blood sample of that, what is the probability that this person is prone to cancer, and what type of cancers he is prone to? If he is having 6 fingers in the hand, what is the reason for that? If he is suffering with chronic diseases, what is the reason for that? Whether father is the reason, or it is inherited from mother, and so on. So for that type of research, and what type of objectives are there for this research? I wish to disable the entire right side of the body. What type of points or type of cells will control the other cell? This type of research was there. So our scope of operating is to identify the protein coding region, not to analyze. So the practitioners or the people from bioinformatics, they will analyze what whether this is prone to cancer or not. All this analysis they will do. But the challenging thing that we have in here is the neighbors that we have. Like when you take a DNA sequence, will be in the terms of AGCTs, AAGGCCT, something like that. So when the challenges that we have for this DNA sequences is like you take a blood sample in the morning, the DNA sequence will change, but the properties will remain same. At this moment, you can say that the neighbor of G is A and the right neighbor of G is C, but at some point of time, if the time over the period of time, the neighbors will change, because the DNA, most of the people know that it is in a helical shape. So the neighbors will change, and slowly what will happen when the neighbors change? You need to have a classifier which predicts something in future. What can be the neighbor of sometime in the morning at 8 o'clock? Have a DNA sequence. If you take the same blood sample at 10 o'clock, you have a different DNA sequence. So the challenges of the research are to predict the neighbors, because the movement of your DNA sequence is a helical shape. And the challenges of research are the sizes of your
Good
DNA
morning,
sequence.
sir. Good morning, sir.
Please.
Sir, your slides are not visible. Is it you're sharing slides, sir, or without slides?
No, slides I have shared. Just I will find out. I will just. I will share it again then.
Thank you very much, sir.
So is my slides are visible, sir?
Now is it visible, sir?
Earlier it
Yes,
was
sir.
not visible.
No, earlier it
So
was
far
not
not
visible.
visible, sir. We are sorry, sir. Thank you.
So sorry for this particular sharing. I thought I have shared, but okay. So we'll just proceed. The objectives of the research we have discussed, and I'm going to the next part. Like the challenges of research are the DNA sequence lengths are different. You need to have a classifier which can handle different uh, DNA sequences of different lengths. And the significance of the research, I said, most of the objectives that we have for mycin will also be attracted to here. Like what will be the disease causing gene if I wish to disable the part, all the significances will be there, and we'll see what are the challenges and significances when you try to implement this. This is what we are discussing for this particular session. So cell automata is an A technique where you can assume that cell automata is a set of cells on a grid. You will have a starting state. You will also have a final state. You can move from one state to other state. If you consider three cell automata like this, if I say you have three cell automata, what is that we can do is three cells will be taken as a state. And if you wish to move from one state to other state, it depends upon the left neighbor here. It depends upon the right neighbor. It can depend also on its state. There should be rules. My intention is cell automata is a innate classifier which was governed by some rules, and that rules can be sometimes I have to depend upon my state, sometimes I have to depend upon my left neighbor, sometimes I have to depend upon the right neighbor, and sometimes I have to depend upon all these three. It depends upon the rule number. So whenever we do some projects in cell automata, we ensure that we have certain rules which can classify the unknown samples to a particular class. The same thing we do. But what type of rules we need to take, how the rules can be taken, that we will discuss. Cell automata is a technique that the intention that we take this cell automata is to see that how do you generate an innate classifier by using cell automata? The answer for that is by the use of rules. So without rules, obviously, most of the cell automata projects will not be helped. So how do you integrate the cell automata rules with particular deep learning models like CNN and RNN? We'll discuss later. So my intention is most of the framework that we have for cell automata will map to the problems in bioinformatics. That is what my claim, and this is what published in a scientific journal by me. So the rule selection plays a vital role in the terms of when you take cell automata. And then we go to the other thing, like the advantage that we have for cell automata is you can go for having this session, having this execution parallelly. Apart from that, no cell will have the overall view of the entire system. This is what the strength of cell automata is. So I said when you are going for interdisciplinary research, the most important thing that we need to understand here is whenever we are working in power electronics, the basics and the particular understanding of power electronics, it's very difficult to understand. The same way when we are working in bioinformatics also, we need to understand the basic logic and basic logicality between the problems. That is the reason I just kept one or two slides so that you will understand the significance and the depth in this research. So everyone will have this DNA, every human body will have this DNAs, RNAs and proteins. You analyze protein, you will get a lot of information like somebody wish to, some doctor has to propose his medicines. without side effects. Then you need to find out the structure of protein. It can be quaternary or it can be triternary. So it's very difficult. Like how do you process DNA sequence? How do you process RNA sequence? It's very difficult. So what is the logicality between DNA and RNA and protein? This has to be known before applying or before executing any of the bioinformatics project. So we started exploring what type of problems are there in bioinformatics, what are interrelated, what should be the first, what should be the next, and so on and so forth. Then we understood that uh, if you analyze DNA, you'll have genes. If you have in DNA, you have genes. In these genes, you have two regions. In these two regions, we call it as introns and extrons. Introns are the major part of DNA which does not have PCR. Extrons are the minor part of genes will have this PCR. So we need to understand the logic between theoretically you need to know what first and what is the next, and what are the structure, and you need to analyze, then only you can apply our AI techniques or this bioinformatics or power electronics and pumps and so on and so forth. So the intergenic regions that we have, we say it is a promoter and whatever it may be. See, if you analyze promoters, you will get information pertaining to the cancer-related diseases. And you analyze introns and extrons like PCR, you will get information about what is the disease causing gene, what can be, who is inherited, what can be the outcome, and so on and so forth. So if you go to the next slide, it will be more clear. Like you have a DNA sequence. If you understood, you can find this pink region, which is an intron, which form the major part of DNA sequence, which does not contain PCR. So the minor part of regions, we call it as exons. This has PCR. That is what the complexity of the project is. And we also understood at some point of time, after a thorough analysis that we have done, we understood that if there is a promoter, it will be at the starting of a DNA. So the logicality that myosin has behind myosin is initially once it processes a DNA sequence in the terms of AGCTs, it try to find promoters. Then if promoter exists only, then PCR comes. So if this, you see the slides, you can contain a lot of biological content initially, but my intention is whenever you wish to apply a particular classifier on this bioinformatics and the power electronics department, you need to understand the domain of the department, then only you can proceed. This is what I just try to have this discussion. Like you will get the promoters here. When there is a promoter, then you go for finding this PCR. This is what I understood after studying a lot of papers in bioinformatics, and the description that we implement myosin initially by using a traditional algorithms. I understood that once the protein promoter region was found, then there is a chance that protein coding region can be given, and then you can just try to find out protein structure from here. So my intention is when you develop a classifier, can this classifier framework can be used to address various problems in bioinformatics or not? I understood that it is, the answer is yes. Majority problems in bioinformatics, whatever the myosin has solved, can be solved by using this structure. That is the reason this work that we have done is benchmarked in the website of SVCW, so that any researcher has done an implementation, he can compare it with our web server, and he can claim that that work can be better or this can work can be better. It is only for just having a benchmark for comparison and doing research in a different dimension. So this slide is very important whenever you are pursuing research in interdisciplinary areas. You need to find out the logicality between various problems, and if you claim that your framework that you have designed will address various problems in bioinformatics, then you can find that this is where this is what the input is, and this is what the processing is, and this is what the outcome that you get. So if you wish me to go to the next slide, then you have a slide on what are the implementing challenges that we have, and why we need to integrate this artificial immune system with cell automata, and what are the challenges that you have, and what is the strength of an artificial immune system, because AIS means an artificial immune system. when integrated with cell automaton, will be a robust and a versatile classifier with considerable accuracy. And at the end of the day, we'll also discuss what will happen if cell automata was integrated with CNN and what type of accuracy difference you get and what type of effect that you get in the terms of execution time. So finally, we understood that the challenges that we have, like whenever you process DNA sequences, DNA sequences of different lengths. Some DNA sequences are of length 54, then some of them are 102, 252, 354. You can see the UCA repository. Now the DNA sequences are of length 102 also, 1002 and more than 1000. So you need to have a classifier which can process all these things. And the cell automata that we have taken is like two-state, three neighborhood cell automata. So every time when it is moving, it needs to consider these three neighborhoods and then it has to proceed. So we thought of developing three classifiers. First classifier is to predict PCR. The second classifier is to predict PR, and this is an integrated classifier which can predict both of them with less time. So finally, the claim that we have made in that SciIndex paper is the third version. Like we have an integrated classifier is a classifier where this is a general framework that can be applied for other problems in bioinformatics also to have a versatile output and versatile and a robust outcome. So we have done, as usual, whenever people are executing these projects, we have done the literature survey and what type of cell automata can be taken and what type of model that we have to use, whether it is a hybrid cell automata or non-linear cell automata. This is what we have done. What are the general cell automata applications that are available? Because cell automata use it for compression algorithms and decompression algorithms, and where exactly the cell automata was used and how the pattern classification was done by the definition of Wolfram by using this cell automata. And what are the applications cell automata with respect to the bioinformatics? What are the other problems that are addressed by using cell automata that we have done the considerable extensive literature survey, so that this literature survey will help us to go into a different dimension of our research. The same way, like we have implemented various protein coding region algorithms individually, and we found that this decision tree and neural network algorithms are better at this moment for the particular enability of particular processing of your large data sets and the input that we got. The same way we have done literature survey of various promising PCR algorithms, and we understood that these two particular algorithms are a very good algorithm that can find out whether the DNA sequence contains promoter region or not. So then we thought of having a considerable literature survey, having done a considerable literature survey, we thought we can go for having this artificial immune system, multiple attractor cell automata. We thought we can take a data structure, instead of a tree, we can construct an inverted tree, because when I assume that you have a tree of high, you have a DNA sequence of 10, and sometimes what will happen is you have to create a tree of height 20 also. But instead of that, what is that we have done is we have constructed an MACA which was inverted like this. So what is our claim is, like whenever you have a DNA sequence of length of 10, you can get the classifier output within the size of size, like within the height of the tree. Whenever height of the tree is 5 itself, we will get the outcome. Yes, we say that this is an attractor basin and all these are transient nodes and whatever it may be. So whenever we thought of designing this artificial immune system, multiple attractor cell automata, this is what the data structure that we have selected. And the ranges that we have fixed at this moment, processing the DNA sequences of length 54 to 354, we got it from UCA datasets and FICIT and standard datasets that we have. And the challenges that we have is we need to process large datasets of order 10 lakh and 12 lakh in together with coding and non-coding sequences. And we need to add a different feature, like we are trying to have a general platform where all this can be kept. So whenever we are trying to have a general platform, you need to understand that this existing features should not be disturbed. At the same time, the existing feature accuracies should not be disturbed by adding a new feature, because we are going to propose an integrated algorithm which can predict both of them. So adding this extra component will help. So obviously this is what the challenges that we have. This is the most important slide that as per this discussion of cell automata is concerned. So this is what I said. Whenever we try to have an innate classifier that has to be developed in cell automata, we need to have some rules. These rules will guide you, like what can be the transitions and what is the outcome of the transition, what type of class it is going at the end of the day. This type of discussion can happen with this. So if you see here, you can understand that if you wish to move from one state to other state, you have to depend upon the left neighbor, you can depend upon the right neighbor, you need to depend upon its state, and so on and so forth. So you have uh, uh, two types of rules. You can call them as complementary rule and non-complementary rule in the terms of bioinformatics. So whenever you think in the terms of implementing the rules, I have taken two rules here, saying that you have a rule called as 51. We call it as a complemented rule, and 238 is a non-complemented rule. Apart from that, you need to understand, like whenever I took a rule like 51, what is that I have written here is the binary number of 51 here. Whenever I take 238, I have taken the binary number of 238 here. So then, since you have considered only 3 cell automata, that means 3 cells are integrated to have a state. So you can have 2 power 3 different type of possibilities, like you can have 0 0, you can have 0 0 1, you can have 0 1 0, you can have 0 1 1, you can have 1 0 0, and so on and so forth. You can have 8 different combinations of neighborhoods that we will get. So you need to find a rule in such a way that this 51, whatever you are writing here, which says whenever you are moving from one state to other state, it depends upon its state. And rule 238 says if you are moving from one state to other state, you have to depend upon the left neighbor as well as right neighbor. Then this is a different type of perception. This is type of a different understanding, because you cannot have a rule for 1 or 2, because 1 or 2 cannot be represented like this. Because whenever you say that rule 1 or 2 exists, you need to write the binary number of 1 or 2 here. And for all the 8 combinations, you have to get the output like this. This is what the combination, this is what the output you have to get. So 1 plus 1, you have to get 0, 0 plus 1, you have to get 1, 0 plus 0. So how do you get this type of output for all the 8 combinations is what, and that type of rules has to be taken. So the overall outcome of this particular slide is you have 2 types of rules whenever cell automata is when you are implementing cell automata. We have complementary rules, you have non-complementary rules. Based on that, you can have this is what the particular you will understand this better. 254 is a rule which states that if you wish to move from one state to other state, it depends upon the left neighbor, it depends upon the right neighbor, it can also depends upon its state. 252 is a rule which will tell you that whenever you are moving from one state to the state, it has to depend upon the left neighbor as well as its state. So 238 says whenever you are moving from one state to the state, it has to depend upon its state and right neighbor. So 252 says it has to depend upon left neighbor as well as right neighbor, and so on and so forth. And rule 204 says it has to depend upon its state, and 240 says it has to depend upon the right neighbor, and so on and so forth. Many rules are there. These are called as non-complementary rules. You'll see also complementary rules like this. Whenever you are applying fuzzy logic for cell automata, you need to understand like your value should be in between 0 to 1. Once the value crosses beyond 1, you will say that it is 1, because if I say if you add 0.6 plus 0.6, if it is 1.2, we will take only 1, because the fuzzy values which were incorporated for this particular project has to be in between 0 and 1 because fuzzy values will take only.
Excellent, sir.
Please, please.
Actually, about fuzzy, what we understand so far from theory and experimental, maybe it applicable to non-mathematical, but whereas you are showing the table is mathematical.
Yes.
I want some clarity, sir, kindly.
Please go ahead.
So here, what my question is, slightly I'm asking.
Please.
That if at all, is there any possibility to define fuzzy rules or fuzzy inference mechanism based on mathematics?
Yes, see, whenever you wish to have fuzzy inferences, inference is very simple. Inferences you can make from some statements. See, I have given some set of statements and you can have some inferences, but the inference is what the conclusion that you are getting at the end of the classifier. But the fuzzy rules that we have applied here is different. The mathematics that I have done here is it will be explained here like okay, this is a better thing. What your question is something like can we apply fuzzy rules to have some inference? Is it right, sir?
Obviously, what your connectivity is correct, maybe what I've been experiencing maybe
It
different.
is right, sir, because whatever you are asking is very relevant to the discussion that we are having here. Because the conclusion that you said, like inference mechanism, we have robust inference mechanisms in fuzzy logic actually. But whenever you say fuzzy,
Yes, sir, please.
whenever you say fuzzy logic, whenever we have some incomplete information and still we need to take a decision, generally we apply this fuzzy logic. For example, I will tell you a small example that I have read recently in an article pertaining to this fuzzy journal. Like we have a murder that has happened somewhere and 4 people are the suspects in that murder, and every person like assume that A, B, C, D are involved in this murder. Every person will have this alibis. Like when this murder has happened, this A was somewhere in a restaurant where CCTV has captured his image, and B say that he was staying in some uncle's house, something like that. So what will happen when your question is like whenever you have an incomplete information, can you take a decision? The answer is yes by using this fuzzy logic. Based on some certainty factors, somebody asked me, do you have rain at 10:30 at Bembaram? The answer is it's 10:30 now. So somebody asked me what is the probability that you have rain at 11 o'clock in Bembaram. If I say the answer is 0.6, that 0.6 we call it a certainty factor. It is the belief that I have, like you have rain at Bembaram is 0.6. The same way, the inferences that you asked is very relevant because inference mechanisms, you have many expert systems. But whenever you say that there is an inference in the terms of probabilities, the guarantee that that will happen is very less, because Rama is a boy is a statement which is valid with the inference 1. But there is a probability that there is a rain at 11 o'clock. The probability may be 0.6 or 0.7 or 0.2. It depends upon the dynamic parameters that will happen. So in this context, whatever we have applied the cell automata, what is that I'm just trying to tell you is whenever we wish to move from one state to other state, we need to apply a rule. Since I have considered 3 cell automata, I am using 2 power 3 combinations where 0 0 will be there. So all these 2 power 3 means 8 combinations I am writing here. And once I write 8 combinations, I have to write the binary number of this 51 here, and for all the rules you have to get this. So the particular project that we have done, we have used many rules. In that rules, these rules will be very easy for some people to understand. This is the reason I'm just having this. So if you just allow me to just proceed, this is what the operation that we are doing. That's what I'm saying. Like whenever fuzzy logic is discussed, the maximum value that a fuzzy logic can have is 1. That is the reason if it cross, like if you have a neighbor with 0.6, and you have a other neighbor with 0.5 and you are asked to add, then you will get more than 1, you have to take 1. So the minimum value that you will get is 0, because all the fuzzy values and membership functions will take the value in between 0 and 1. So this is how we try to represent our particular rules in the terms of the classifier that we are going to generate in the terms of cell automata. Like I said, we have taken 3 rules here, 254 and 85 and 40. 254 says whenever you are moving to from one state to other state, it depends upon the left neighbor and its state and the right neighbor, which will say that this is what my matrix is. And 85 rule is the second rule which was standing here. 85 is standing here. It says it has to depend upon the right neighbor, so we have kept here. And 240 rule is staying here. And 240 rule says it has to depend upon the left neighbor. Left neighbor is this. That is the reason 1 it was there. And we have to take other matrix. This is what the implementation that I'm just saying. You can have your own implementation. This is what how you implement and was done our work actually. So this one says 85 is a it is a complement rule. This is what it is. So you can have your own implementation, but this is how we have proceeded by using this encoding and decoding. Whenever I am doing this particular rules, whenever I'm going for encoding and decoding, this type of matrices we have used. So 1 says in U matrix says that it is a complement rule, and 0 says it is a non-complement rule, and T says it will give you the dependability of the particular rule. So if you proceed to the next slide, this is how the rules are applied, and this is what the sample input was given. Like I have taken the standard input from Fickett and Dung, like 54 length data set I have taken. In this 54, I have only taken these 3 particular locations that I have taken, and I have applied these rules that were discussed now. So when this rule was applied, this rule says whenever you are moving from one state to other state, at this moment you have to depend upon the right neighbor. So the right neighbor is 0.2, so 0.25 come here. So again, the rule was applied to the second rule here. The second rule says it has to depend upon its state and right neighbor. So its state is 0.7, the right neighbor is 0.5. 0.7 plus 0.5 is more than 1, so we take only this 1 here. And so on and so forth. We will move from one state to other state, that you will not get other state here, so we'll stop here. And we call this as attractor basin, and this attractor basin will have some property. This is the basic operation of cell automata. We call that as an attractor basin that will have a property whether it has a coding or non-coding. I have taken a 3 state cell automata with only 3 locations. This much complexity the diagram is now. The same way, when you apply a different rule in the terms of when you use a complementary rule, this is what it is. You apply the rules and this is what the transitions that you get. And sometimes the complemented rules you will get the output faster. Sometimes in the combination also you will get. And there is no rule that you have to use only 3 set of rules or 4 set of rules, but we have used 3 set of rules. The reason is the way in which you are applying it with 3 in the terms of codons. That is the reason we have used this, and the other reason for this is we have considered 3 cells as a state. That is the reason we are going for that. So this is the general system architecture we have represented here for this particular presentation. Like we took standard data sets from Fickett and Datasen, and apart from Fickett and Tongue data sets, we have also taken the data sets from UCA for various other processing, other particular domain related instance. Like the input was processed in the terms of 3. The reasons is codons and exons that we have seen earlier were in the 3 positions. Apart from that, this 3 cell automata was used. So every state will have 3 cells. So we have seen what exactly is a rule generator is. We will also see how this construction of basins will be done by using a small example in the next slides. This is what the training algorithm that we have for this particular session. All these training algorithms are same because I said we are using a 3-state cell automata. That is the reason we are encoding the 3 things in the terms of cell automata, and the real attributes like where exactly this A was located in this particular frame of 0 to 1. Is it 0.4 or 0.3? This is what we got it from the website itself. So then the rules will be applied from this root generator, and we'll find out how this AMACA was designed, and then we'll see what exactly, how many iterations will be done, what type of population you have to done, and what type of crossover and mutation will be done. This is what the testing strategy that we have used. And this is what a sample data set that I have taken from Fricket and Tongue to find out AGC if it is there, and this is where the AGC are located, and were given some classes here. And 6 attractor basins were given, and this each attractor basin will have its own class. You can say that ABC are coding or DEF or non-coding. It depends upon the input and output at that time. And fuzzy values were already were also given in the terms of data sets, where A is there 0.2 and where B is there, and you need to find the location of your PCR, and the analysis part will be done by the practitioners. A small input of size 1, 2, 3, 4, 5. So you take 5 inputs. This much complexity you will get if you apply this particular rule called as 170 and 254 and so on and so forth. Slowly just proceed to the next slide. You have a other complementary rule that we have, and here if you apply the same input with a different rule, this is what the complexity that we get. So you need to assume, like when you have an input of almost 252 length linear sequence with 252 length sequences that were given and the location that were given, how much LCT that you will get, you need to identify and just find out. And I just kept more images so that you will understand what exactly the transitions and how the transitions are happening. But here, based on the basis that we have, this basin will have a property, and the entire structure which was coming here is falling into this, and this 11106 is a basin, 11106 is a basin which was so this will have a particular property where we need to find out where this F is. F is coding or F is non-coding. That F it was here as per the data set that we have taken. F is pertaining to a class 2, and we say that class 2 is non-coding. This is how I represented actually I have taken a raw data set, and through images I understood, I just try to explain whether this will be coding or this will be non-coding, something like that. So this is how an MACA basin looks like. So how can we create, and what is the essence that we have in the terms of inverted data sets, and how these elements will be placed in this attractor basins and so on and so forth. And this is what you need to assume in the terms of inverted tree. And this is the criteria, like whenever you wish to have a particular rule that is applied, when you find out the fitness function, how many are classifying correctly, how many are not classifying correctly, this is what we have to do. because we're using an artificial immune system, we are having this antigen nomenclature to have a better rule and better population every time when it was these rules are every time were classified based on the fitness. So by the end of the day, when we are reaching, whenever we have reached to the next level, we have almost top n promising rules that can classify every input to the next level. So this is what you have a modified classifier that was proposed by us. There's no need that we have to discuss. But this is what the implementation things that may be useful for some of the people who wish to work on the cell automata and deep learning integrated structure. So the basic nomenclature that we apply again here is like we take a rule and we try to have a different rule with a different type of fitness which can be benefited at some point of time. So here I have taken a rule, and this rule was represented like this because 1, 3, 2 are the encoding strategies that we have, and the same set we have taken that 1 is a complemented rule and 0 says it is a non-complemented rule. And you apply some mutation is something like a flipping the values so that at the end of the day the new rule was framed and we try to find the fitness of this rule, whether this rule is better or the old rule is better. Based on the fitness, we'll keep the new rule or we'll just remove the new rule based on the fitness. So I will not take more time. Like I will come to the particular results that we have. Like what happened. Like what type of data sets we have taken and what is the amount of data sets that we have taken. Like I said, it will be more than 7 lakh data sets that we have to process of different sequences, because DNA sequences are of different lengths, like 54 and 108, 162 and 252 and 354 and so on and so forth. Different lengths DNA sequences, and we need to understand that this different DNA sequence, this classifier has to process this DNA sequences of different lengths, and this is the amount of number that we have to process and find out the adaptability and usability of your classifier. The same way for this PCR also, we have to take this number, and total it can be around 8 lakh data sets that we have to take. And since I said it is a multiple attractor cell automata integrated system, you can just try to find out, you have a different tree. 10 minutes per we will just discuss later. So you have a AS-MAC tree with different types of same length 54 base pairs and have 4 best trees here. And we have used the set of how many futures that I have used. Based on that, what is the accuracy that I have, and based on this 4 best trees, whenever I have a length of 54, I can classify and get the accuracy at least 92. Based on the usability and based on the number of futures, if I have 4 futures, if I am getting 92.6 average, and because I'm not able to classify, I have to go for more number of futures to classify the things. So this is the best tree that we have got for a length of 354. Some futures I have kept intentionally. A Fourier future was used, and position receptor was used, and many other rules are used, and because futures are also used. And there are different type of parameters that we have used to say that this is a better project, or this is compared to other things, this is better, because we have the standard nomenclature that we have from the literature survey is having all these parameters. We felt this parameter has to be considered to compare our work with the existing work. So this is what our work is, and this is what the sensitivity and specificity and accuracy that we have for this 54 length data set. Similarly, 1 or 2 we didn't have. So different type of length sequences, we have a different type of slide that I have kept, so that this type of data sets or this type of lengths we have to consider. That is what my intention is. So this is the performance comparison that we have done for this existing system in the terms of 162 length base pairs, and an artificial immune system based cell automata will have an average accuracy of 92.45. At this moment, I used to say that when this AAS-SMACA was integrated with RNN, we are getting an accuracy more than 95 with 0.7 nanoseconds. So this is what the entire aim of the discussion, like whenever the cell automata is integrated with deep learning models, sometimes what is that we are getting is we are getting accuracy also increased, and we are getting the output faster than what we have done with a single cell automata classifier. This is what I wish to compare the promising existing systems as well as decision trees and neural networks with this AIS MAC system, and this is what the fact that we have got. And this is a simple representation that I have made this PowerPoint for this particular FDP. And apart from the specificity, sensitivity, and other classifier accuracies that we have represented, we also represented the memory overhead based on the way in which you build this attractor basin in the terms of intermediate nodes and so on and so forth. And then we have compared this memory overhead with the other classifier that we have prepared also, because we have an integrated classifier, we have a classifier which predicts this PR, we have a classifier which predicts PCR, we have an integrated classifier which can predict both of them. So this is what the classifier time, and this is the fuzzy state that we have whenever the implementation has happened. Finally, we have arrived at the common platform, but this platform, we claimed that this is used for addressing these two problems like PCR and PR. These are the few signal features and context features that we have used for having this particular PR, because PR is more important than PCR. This reason is, if you analyze promoter regions, you will extract a lot of information regarding the cancer possibilities. This is how you implemented. This is the capital C's and T's. I will give you where exactly this promoter is identified. And these are the amount of data sets that we have applied this for this particular features. The best AS PRSTs were located for this 252 length or 251 length base pairs. And again, the many parameters we have considered for having this discussion. So just a minute, just. So all the standard parameters are considered to process this, and this is what the integrated mechanism for identifying both of the regions, where in this integrated system we try to find out the first boundary, and then based on the first boundary we are going to find the next boundary. So what is that we have done is we have found this promoter region first. Then from this promoter region, we have identified this PCR, and we have claimed that if both of them are integrated, we are getting the output with 0.7 nanoseconds. This is what our claim, and this is what the paper that was published in our scientific journal. This is what the amount of complexity, the amount of depth that was involved, and these are the major contributions. Like the major contribution that I can claim is like we have prepared, we have identified a platform where most of the problems in bioinformatics are can be solved, and we have done a very good literature survey. That's what I said. Conclusion is we have different type of classifiers for PR, PCR, and PR, but we have an integrated algorithm for having both of them, and general framework was developed. Now we have developed the general framework with cell automata and deep learning, where if we have a chance, I will take other session in the evening after Dr. Mishra madam's presentation, so that we will discuss about what exactly will happen if you integrate cell automata with deep learning. And this is what the future extension that we have done. We have already implemented this part, like what we have used a cell automata integrated with deep learning mechanism to predict the structure of protein. This is what we have done, and that paper was published in a Scopus Index Journal. This is the number of references that we have made. And you have any particular discussions that has to happen, we can discuss, or we can just conclude this session here and we can join at 12 o'clock later.